Okay, let's go ahead and start. So before we do start, just I wanted to put up I think an interesting program for those of you who are involved in research and how to deal with the stresses involved in research. So if anyone's interested, and then you have a conflict between going to the review session that's tonight, so that's my way of reminding you that there is an ad hoc review session tonight from 5:30 to 6:30 that's competing with this. Don't worry, there are there are other review sessions that we can deal with, both laboratory and lecture material. So don't feel too conflicted if you wanted to go to both of these and we're having a hard time making up your mind. Okay, so on with our science program. No more public service announcements today. We left off talking about how influenza virus makes certain that its messenger RNA can be translated. Because remember that influenza virus, a member of the orthomyxoviruses, has negative sense RNA. So it has to first be turned into positive sense. And so we talked a little bit about this cap snatching mechanism, or decapitation, as some people refer to it, that allows one of the viral replicase subunits, PB2, referred to here as viral endonuclease, to cleave host cell messenger RNA in the nucleus. So this does two things. It's basically eliminating the host cell from translating its own messenger RNA, and then hence making an antiviral effort to try and stop the virus from replicating. So that does one good thing from the virus's point of view. But the other thing that it does is that it takes this 5' end of that messenger RNA, including that 7-methylguanosine cap, and now uses that as a primer so that the other subunits of viral polymerase, which came in preformed at the time of infection, will now use that host mRNA, just that little snippet of mRNA, as a primer to begin to synthesize an RNA copy of the RNA genome. And remember, by definition, that's what viral replicase is. It makes an RNA copy of an RNA genome. Now, it's a little bit more complicated even than that, because what you're going to see is that this viral polymerase, viral replicase, actually makes, at different times during the infection, different species of positive strand copies of this negative strand. So at the point that we're at now in the pathogenesis of influenza, we're very early in the infection. So infection has just occurred. This piece of negative sense RNA genome has just been imported into the nucleus of your tracheal cell, for instance. And the first thing the virus needs to do is to begin to express the protein that's encoded on this random RNA segment. So the first type of viral replicase activity involves this cap from host messenger RNA, and then viral polymerase makes an RNA copy of the RNA genome, but it does not make it full length. So this is going to be the key point in the difference between the two types of positive strand copies of this negative strand that viral polymerase makes. Early on in the infection, RNA polymerase is primed by this cap from the host mRNA, and then the RNA synthesis stops at a polyadenylation signal. So a stuttering will occur at this particular point, and RNA polymerase, the viral replicase, will stop the synthesis of RNA at this point and put a poly A tail on. Now, this messenger RNA just made is now perfectly synonymous with any host messenger RNA. It looks just like your RNA, messenger RNA or mine. It has a poly A tail on the 3' end. It has a 7-methylguanosine cap. This RNA can now be and will be transported into the cytoplasm of the cell to be translated into whatever the protein is that this particular RNA segment encodes. But recognize that this particular transcript has not gone the full length of this negative strand. So it did not make a positive strand copy of that full genome segment. That's going to occur later in infection. At this point in infection, the only thing the virus is concerned with is making messenger RNA that can be translated by the host to make viral proteins. So that's what's occurring first, using this cap snatching method and then this polyadenylation signal. Here's just another way to look at it I stole from another textbook. Here is the viral replicase, the PB2 subunit, B standing for basic. P, you can always remember, stands for polymerase. There are two basic subunits, and that's what the B stands for. But the replicase will cleave the 5' cap from this and now use it as a primer. This host messenger RNA that's left behind is useless. It will just be degraded, perhaps, but it certainly won't be translated or transported. Viral replicase will now make not a full length, but it will make an RNA copy of that viral RNA. This was the genome segment that came in, one of the 8 genome segments that came in. And now you have this polyadenylated on the 3' end piece of messenger RNA, 5' methylguanosine cap, perfect replica of a host messenger RNA bound to be translated. So these are, again, the 8 segments of the viral influenza A virus. And so we've talked about the fact that, these different RNA fragments are, for the most part, encoding a single protein product. That's not entirely true, and it's even more complex than I've already given you to believe in the first lecture. But for the moment, I want to reiterate the fact that segments 7 and 8 actually encode 2 different proteins, depending on which form of RNA is made from segment 7. So a full-length transcript made in the nucleus of influenza segment 7 will, in the cytoplasm, be translated into the matrix protein 1. So matrix protein 1 is part of the nucleocapsid. It's a structural protein. At some other frequency, that transcript from segment 7 will actually be spliced. An intron-like element will be removed from it. It will be then transported into the cytoplasm, and now that shortened section will be translated into a completely unrelated protein, different protein amino acid structure. And that's going to make that matrix 2 protein, that's the proton channel that allows for the acidification of the virus early on in infection. Same thing with segment 8. Segment 8 can actually be translated from a full-length transcript, or I shouldn't say a full-length transcript, but an unprocessed transcript of segment 8. That unprocessed transcript will be translated in the cytoplasm as non-structural protein 1, but at some frequency, the messenger RNA that is made of segment 8 in that nucleus will have another intron-like segment removed from the RNA. The RNA is spliced back together, and that altered or that spliced form will now be translated in the cytoplasm into a completely unrelated protein, non-structural protein 2. So these alternative splicing actually adds to the complexity of the proteins produced by this virus. But still, at this point, we're only talking about 10 proteins produced by influenza. In a few minutes, I'll tell you about the fact that there actually are more than this, things that have been recently discovered in influenza. So there's alternative splicing for just 2 of these forms. The matrix 2 protein, and then in terms of where these proteins end up, and we have a much better figures for this, I'll show you a little bit later in the lecture, but 3 of these proteins are going to be trafficked to the cytoplasmic membrane. So when the proteins, matrix 2 protein, the hemagglutinin, and the neuraminidase are translated, they're going to be translated into the endoplasmic reticulum, transported via the Golgi, and end up trafficking to the cytoplasmic membrane, because these are the proteins that are going to be in the envelope of the newly synthesized viruses that are going to bud off from that cell. So we're going to get to that in a little bit. I kind of got ahead of myself a little bit. This is just another way to look at these splicing models and to show you that it's even more complex than that. So here is segment 7. So segment 7, messenger RNA, if it's made in its entirety without any splicing, will be translated in the cytoplasm. I believe this little thing is meant to represent a ribosome. I'm not really sure. But in any case, the M1 protein will be translated from the unprocessed form. But at some frequency, that RNA that's made by the viral replicase will have a region spliced out of it, so that now a region that could not previously be translated is in fact now translated. And that will result in the matrix 2 protein. Same exact process occurs for segment 8 that encodes both the non-structural protein 1 and non-structural protein 2. You can have non-structural 1 created from an unprocessed transcript, but the processed transcript, the intron element removed, will be translated into a completely different protein. There's a downstream of this stop codon for non-structural 1. There's another start codon which will encode this other non-structural protein. So that shows you how you can get 10 proteins from 8 segments. But it's actually a little bit more complicated than that. And this is something that we really didn't realize until probably about 2001 or 2002, when a famous influenza researcher recognized the fact that within the segment of RNA, I think it's segment 2, that's not important, that encodes PB1, so part of the polymerase, one of the basic subunits, PB1, actually has alternative start sites within it. We won't talk too much about this PB1N40. What this really is just a slightly truncated portion of this part of the polymerase, where the first 39 amino acids are not translated. But there's an alternative start site that can be recognized by a ribosome. So a ribosome can recognize this single piece of messenger RNA coming out of the nucleus complementary to segment 2. If the ribosome starts translation at one start codon, it will make the full length 758 amino acid polymerase subunit PB1. But at some frequency, due to something, a leaky scanning mechanism, this ribosome can start at another start codon downstream by 39 amino acids. But it will start there and make this slightly shortened, an amino terminal truncated form of PB1. But the amino acids are exactly the same between these two. There's no frame shift here. So PB1N40 is exactly the same as PB1, just missing the amino terminal 39 amino acids. I'm not going to say anything more about this particular form of the protein, and you can correctly translate that as, and I don't know what the hell PB1N40 does. I'm not sure that other people do, but I bet they know more than me. The more interesting one and the more impactful one is a third alternative start site, which has a different reading frame. So PB1F2, F standing for frame. This is a +1 frame shift. So there's yet a third start codon, which is present in some, and this is the important point, in some viruses, but not all. In viruses that have this third start codon in the +1 reading frame, they will create a small peptide called, appropriately enough, PB1 frame 2 or F2. Because it's in a different reading frame, it shares virtually no amino acid homology with the protein of PB1. Yet it's encoded from the same piece of RNA. The really interesting thing about PB1F2 is that it is found in every influenza A virus that infects birds, and it is never found in any of the viruses that circulate in mammals, except for the 3 pandemic strains that we've been able to characterize: the Spanish flu, the Asian flu, and the Hong Kong flu. The 3 major pandemics of during the last century all had a different version of segment 2 in their genomes such that they could produce PB1F2. And that's actually one of the major keys as to why those viruses had specially high virulence. This is a form of PB1 that's normally found in birds, not found in humans. But this little protein, this only about 100, it varies in size between isolates. But this PB1F2 protein actually targets the human mitochondria and induces apoptosis. So this is part of the reason why these viruses are particularly deadly and particularly virulent, is because they're producing a peptide that the normal influenzas that you and I have been infected with don't produce. And so by targeting the mitochondria and depolarizing the mitochondria, they induce the cells to go through programmed cell death or apoptosis. So this is believed to be one of the major reasons why the Spanish flu, for instance, was so particularly dangerous. But the Hong Kong flu and the Asian flu later, that didn't kill nearly as many people, also had PB1F2. And as I'll show you in a few minutes, one of the reasons why the pandemic that you survived in 2009, the swine flu, one of the reasons it wasn't more severe is that's the one pandemic that we know of that the virus did not possess PB1F2. That's potentially, or at least a lot of people, influenza researchers believe, that's potentially the major reason why the swine flu was not as lethal, not as virulent as it could have been. So really fascinating. So here's an 11th protein and possibly a 12th protein that influenza can produce. But they do it not by alternative splicing, but by using alternative start sites, by having the ribosome scan for a start site slightly differently. This PB1F2 is still a source of a lot of people's research careers at this point. And so this isn't part of your PowerPoint slide because I didn't want you to get to know any more really in terms of an exam than what I had put on that last slide. But from a recent review I found is that people have now got experimental evidence that this small little peptide can increase the frequency of secondary bacterial infections in animal models that are infected. And this is very similar to what happens in humans. In reality, influenza, the viral infection, doesn't kill that many people directly. What it does is it allows for secondary bacterial infections of the respiratory tract that wouldn't normally occur. And it's those secondary bacterial infections that turn lethal, especially in elderly people. And the presence of this little peptide in animal models increases that secondary bacterial infection. It also has tremendous effects on inflammatory cytokines. So you may have somehow or sometime heard the term cytokine storm. So one of the things that people have hypothesized about why young people were selectively, started to say targeted, they weren't targeted, but younger people tended to die more frequently from the Spanish flu in 1918 was because they had such a robust and healthy immune system that a virus that had this PB1F2, by now dysregulating that immune response, that really healthy immune response that each of you have, actually turns against the host and causes much greater inflammation than would happen in somebody, say, my age. And so that's potentially what caused that very unusual death rate or distribution of people dying from that was that the PB1F2 affected the immune response. So it's a fascinating protein, and the jury is far from out on this. As I said, it's only been discovered in this century. Nobody knew that this existed. Okay. Now, moving on a little bit further into the infection, and I have a nice image from your textbook a little bit later in here where we'll go through the entire infectious process from beginning to end in one figure, which I think is one of the great figures from that textbook. But before we get to that, I just want to tell you that because I already set up my story that this initial transcription that's occurring early on in the infection is based on the cap snatching activity of PB2 and then making an RNA copy of the RNA genome that's not full length. Well, eventually, Later on in the infection, when proteins have been made and the virus is ready to escape that cell and to infect other cells, there needs to be a change in the program of the viral polymerase. It needs to now stop two things. It needs to, first of all, stop using this cap from a host messenger RNA as a primer, because otherwise the viral RNAs that are going to be packaged and put into the next virus are going to always carry a little bit of the host genetic sequence with them, and that's not part of the plan, if you will, for viruses. But the other very important thing is that the viral polymerase needs to stop recognizing this polyadenylation signal and to make a full-length copy of this genomic fragment, this negative sense. Because otherwise, if it doesn't do that, it loses this portion of the sequence to go into the next to the next virus. So the signal that changes the viral polymerase from this initial stage of using a host cap as primer and recognizing the polyadenylation cycle signal is the fact that these proteins, the viral proteins that are now being transported from the cytoplasm into the nucleus, now reach a critical point. So proteins like the non-structural 1 and 2, matrix protein 1, and the polymerase proteins PB and PA, all made in the cytoplasm and now transported back through nuclear pore complexes. Once they get into the nucleus and reach a certain concentration, they actually change the way that viral replicase will replicate this negative strand. It eliminates the presence of these proteins at high concentration, eliminates the need for this host cap, the snatched from messenger RNA. Now the viral replicase will actually make a copy of this negative strand without using a primer. No primer involved. Polymerase will bind on here and begin to make an RNA copy of that RNA genome and will now ignore the polyadenylation signal and synthesize an RNA copy of the full-length portion of that genome, something that's referred to as C, or copy RNA. This could also be referred to as the antigenome. So I use the term antigenome in poliovirus because it's a commonly used term in poliovirus. I don't know if the term antigenome is used in influenza biology, but in essence, what's being made now late in the infection is akin to the antigenome that's made in poliovirus. So these now are turned into full-length positive strands, which can turn around and be the templates for viral replicase again to now make the full-length negative strand. Because remember, what's going to be packaged and put into the viruses, what must be packaged and put into those viruses, is single strand of the negative sense RNA. Viral replicase, as it's shown here in this slide, is making positive strand. Remember, when you make a copy of negative strand, by definition, it's positive strand. And then, when you turn around and make a copy of that positive strand, by definition, that single strand copy is negative sense. So the viral replicase needs to copy this full-length cRNA in order to make the negative strands for packaging. So now, once those ribosomal particle or ribonucleoprotein segments are synthesized and put together in the nucleus, they're going to be transported out of the nucleus, where they're going to go to those portions of the membrane that have been targeted with the hemagglutinin and neuraminidase. So remember, we talked about the fact that 3 proteins need to be inserted, 3 viral proteins need to be inserted into the host membrane so that the virus, as it acquires the envelope in leaving that cell, is going to have the hemagglutinin protein, the neuraminidase protein, as well as the matrix 2 protein, that ion channel. Those glycoproteins have all been localized in rafts or patches at a particular portion of the cytoplasmic membrane of that respiratory cell. It's just sitting there idling, waiting for the ribonucleoprotein complexes to come out of the nucleus And so now you see here the matrix protein, the matrix 1 protein, assembling just below the lipid bilayer. And then within that are the 8 different segments, these negative sense strands of RNA that will have, although it's not shown in this particular cartoon, these will have associated with them the nucleoprotein as well as the 3 subunits of the viral replicase, the PB1, PB2, and PA proteins. They're all associated with that. So now all of these proteins are present. The virus can and will bud off from that cell. So the viral capsid is shown here from the matrix protein inward, and then this little bubble peels off, and so the virus is now released from that cell as an enveloped virus, potentially ready to infect another cell or another host. So here is what I think is a really wonderful slide that shows the entire process from beginning to end of the influenza virus life cycle. So let's just go through this, starting with step 1, viral uptake. So the virus here, the envelope virus, is going to bind to a receptor that contains sialic acid on the membrane of that cell, and that receptor or that binding is accomplished by the hemagglutinin protein. Now that will trigger receptor-mediated endocytosis. The virus will now be taken up into an endosome, shown here in step 2. And now the host cell recognizes that there's something bad within that endosome, and it tries to kill it. And usually acid is a pretty good means to kill foreign invading organisms like bacteria. They don't do well under acid. So your respiratory cell pumps in protons, but actually that's the worst thing that it could do, because these protons are now going to change the protein conformation of that hemagglutinin. So the protons are going to get access to the interior portion of this virus through this little green thing here. That's the M2 protein channel. That's going to change the conformation of the hemagglutinin molecule, which is now going to cause the fusion of the viral envelope and the endosome membrane. And that's going to release all 8 of those ribonucleoprotein complexes of viral RNA. So those proteins actually have nuclear localization signals within them that are only exposed when the matrix protein 1 is gone. But these ribonucleoprotein complexes will now be transported through nuclear pores. What's not shown in this picture, so this picture's not perfect, what's not shown in this picture is that each one of these RNA segments is also associated with the 3 subunits of the polymerase, the viral replicase. So those proteins are going into the nucleoplasm as well. So that negative strand of RNA, each of those 8 ones that comes in here, is now going to be transcribed using that cap snatching mechanism. So here in the center, these tan-colored spheres with a C in them, C standing for cap. These are the host caps snatched from messenger RNA by one of the subunits, the PB2 subunit. So now some of these positive strand copies, partial copies, will now be transported into the cytoplasm, where they can be translated into proteins such as the PB1, PB2, PA. Some of these messenger RNAs are going to be transported into the cytoplasm, but they're going to be trafficked to the endoplasmic reticulum. So things like the hemagglutinin protein and the neuraminidase protein are now going to be put into the endoplasmic reticulum. The same thing goes for that third glycoprotein that needs to go in there. That's the M2 protein. And remember that the M2 protein is made from a spliced form. So the M1 shown here at the bottom of these messenger RNAs needs to be at some frequency spliced into an alternative form that gets transported out in order to synthesize or to be translated into membrane protein 2, that very important proton channel. So those 3 glycoproteins are now going to be synthesized into the endoplasmic reticulum, trafficked through the Golgi, and they're going to be assembled into these rafts or patches of membrane. So by means that I don't understand, most of the host proteins are removed from these patches, and most, if not all of the protein in these particular rafts is either hemagglutinin, neuraminidase, and to a smaller degree, that matrix 2 protein. So this is the patch that's sitting there waiting for the nucleocapsid to come and assemble from in there. But, so what happens next is that these other proteins that are being synthesized in the cytoplasm, the non-structural proteins, the polymerase proteins, they actually are secondarily transported back into the nucleus, the nucleus of the cell. And when they reach a particular frequency or concentration, they're going to change the program by which viral replicase is making copies of its genomic RNA. It's going to stop making these capped messenger RNA segments and instead is going to make a positive strand copy, full length, but without the cap. So what's happening here in step 8 is that original negative sense strand is being transcribed into a full length positive strand, and it's being coded, or it's shown here as coded with nucleoproteins. But then that positive strand is now, or the antigenome, or cRNA, shown here at 8, in step 9, is going to be transcribed again. So a negative sense strand is going to be made from each of those positive strands that's now going to associate with all of the proper proteins, the matrix 1 protein, as well as the nucleoprotein. And then that ribonucleoprotein complex is going to be transported out of the nucleus and is going to assemble into a nucleoplasmid, or nucleocapsid rather, at the membrane. So now it's going to be pinched off from the cell, and then with the activity of neuraminidase, it's going to be released. Remember that neuraminidase plays a role at the release point, because what can happen is as this virus buds off with its hemagglutinin molecules, sitting there waiting to bind to sialic acid containing receptors, it's actually leaving a cell that has nucleic acid, has sialic acid containing receptors on it already. So the virus can get tangled up, if you will, on the surface of the cell that just produced it. So neuraminidase cleaves the sialic acid off of those receptors and thus frees the virus to float away, to either infect a neighboring cell or to be coughed out or spit out or whatever, to potentially infect a naive host. So the virus is setting itself up for success. But here's where the virus is really, so this is a very complicated virus, and it is very highly evolved, and it's a very successful virus. But it's actually highly inefficient as well. This segmented genome that we're showing here as putting 8 segments into these viruses, actually that occurs very seldom, because the virus has no active means to ensure that as viral particles bud from that cell, they have all 8 segments. And if they do have 8 segments, they may have a duplicate of segment 4 and be missing segment 9 or 8. So in reality, the vast majority of the viruses that bud from any one of these infected cells are defective particles. They can't accomplish the next infection. This is part of the reason why these viruses are so prolific. So many viruses are produced from any one infected cell because they have to be. Because the majority, only one, mathematically figured out anyway, only one out of 416 viruses that are going to be released from that cell are going to have all 8 and the proper 8 of these RNA segments. So highly, highly inefficient, and that actually works against us, because this virus has to produce so many viruses from each one of our infected cells that it's such a stress on our cells, we lose so much membrane material with each one of these envelopes of a virus that buds off, that's part of the stress that actually ends up killing these cells. They would probably die from not having their host, their normal RNA synthesized anyway. But this is a tremendous stress on the cells. So very, very inefficient in terms of putting together infectious viruses. But they're not quite infectious yet. So this is a really key point because it also comes into play in terms of why it is that you and I are less likely to get avian flu, bird flu, from a person than we are to get another case of human virus. Because that hemagglutinin molecule needs to be processed. And it's processed in order to make it infectious by an enzyme or an enzyme activity that we have natively. So as it turns out, our cells, our respiratory cells, as well as the secretions that bathe our respiratory tract, have trypsin-like activities. So trypsin is the enzyme that you're all familiar with that breaks proteins down. It's used in digestion. But trypsin-like protease activity is necessary to cleave a hemagglutinin molecule into two molecules called hemagglutinin 1 and hemagglutinin 2. So this trypsin-like activity associated with our respiratory tracts will cleave the hemagglutinin either as the virus is leaving that cell or perhaps before it infects another cell. But it has to occur. So what happens is that the trypsin will break a peptide bond between HA1 and HA2. So the proteins are now actually two distinct proteins, but they don't dissociate from one another. They're held together by a cysteine-cysteine disulfide bond. So they still maintain an interaction between hemagglutinin 1 and hemagglutinin 2. The reason that cleavage is really critical is because without that cleavage, the acidification will not change the conformation of the protein. This breakage between hemagglutinin 1 and hemagglutinin 2 gives that protein enough flexibility that when it's exposed to high levels of protons, acidity, it can change from a conformation like this, where you see that the hemagglutinin molecule 1 is bound to a sialic acid containing receptor on the target membrane. And the other portion, hemagglutinin 2, is still embedded in the viral envelope, shown here at the bottom. The protein conformation that occurs under acidity or low pH, and again, this can only occur if the protein has enough flexibility due to the trypsin-like cleavage of HA1 and HA2, that conformation change that occurs is actually going to take a portion of that hemagglutinin molecule called, appropriately enough, the fusion peptide. Here's the fusion peptide here at pH 7. It's existing free outside of the virus and outside of the target membrane. But under low pH, that conformation change literally throws that fusion peptide into the host membrane. What that does is that brings the two membranes so close together, kind of like two soap bubbles, that when they touch one another will actually fuse. So that proteolytic activity is absolutely crucial to allow these cells to be infectious. And the reason why this is important for you to understand is this is part of the reason why we don't have very frequently, if at all, person-to-person transmission of bird flu. So probably since this H5N1 high pathogenicity avian influenza came to the fore sometime around 1998 or maybe a little bit later than that, there have been probably about 600 cases or more, probably more now, but at least 600 cases of humans being infected by H5N1, and more than half of them died. I mean, so this is a serious, serious infection. This is an infection that you don't want to get. Most of the people, if not all of those people who were infected, got it from being in close contact with poultry. And so these people probably got a major lungful because they worked with poultry that were infected. Fortunately for us, those people who were infected, unfortunately for them, more than half died. Fortunately for the rest of us, there was very little spread to family members or healthcare workers. I think it did happen a few times, but it was very rare. Part of the reason people believe that as yet H5N1 is not capable of spreading from human to human, has to do with this proteolytic activity, this trypsin-like activity. This hemagglutinin number 5 is not a hemagglutinin that can normally infect humans. Humans can only be infected by viruses. There are 16 different kinds of hemagglutinin known. All of them infect birds. Only H1, H2, and H3 viruses with those hemagglutinins will infect humans. H5 will not normally infect humans. It infects them under this very rare circumstance where people get enough of the virus from birds. It's not spreading from human to human because you and I, fortunately, don't have the proper proteolytic activity to cleave this form of hemagglutinin. So your cells in the lung that can produce, that get infected with this virus from a bird, those viral particles were probably pre-activated by the bird. The birds have this proteolytic activity that has the capability of cleaving HA1 and HA2. So that virus, if it finds a receptor in the human lung, and it does, sadly, can infect that lung cell. And that lung cell can produce virus. But that virus is very unlikely to infect anyone else because we don't have the proteolytic activity to cleave that form of hemagglutinin into HA1 and HA2. So that virus is kind of dead end for us. So the worry is that that this virus will actually acquire a mutation in its hemagglutinin that allows our proteolytic activity to cleave it. If that happens, and it might not take that many amino acid changes for it to occur, but if that happens, then potentially the only barrier that's stopping this, 50% lethality virus from spreading to human is gone. And this is not simply hypothetical. This exact same situation happened in veterinary science not that long ago in this century. There was a major outbreak of canine influenza in greyhounds, racing greyhounds in Florida in around 2002 or 2003, with just a tremendous mortality rate. It turned out that virus was a new virus. So dogs have their own influenza virus. But the virus that was killing these dogs was actually 100% horse. It was an equine influenza virus that had acquired just enough of a mutation that it could not only infect dogs, but it could spread from dog to dog. So there was a huge worry for a long time in the veterinary, in pet owners too, that this was going to wipe dogs out because the initial reports had like 50 or 60% mortality rate among dogs. Turns out that the mortality rate's not that high. It's still a very serious disease. The fact that it was in racing greyhounds, which sadly are not that well treated, and whose immune systems are not that good, that artificially inflated, I believe, the mortality rate. But it's still a problem. But this is a lesson for the fact that what we worry about happening with bird flu jumping to humans has happened. We've actually witnessed it happening, just coming from horses and going into dogs. So it can very easily happen to us. So here's this. So this slide from the textbook actually shows that proteolytic activity. It's tending to show, at this particular case, that the protease is occurring, it's cleaving that hemagglutinin just prior to the infection. I think there's actually 2 points in which this host protease can activate this virus, either as is shown in this particular slide, where the hemagglutinin is being processed by this host protease at the start of infection, but I also think it's very important at the end of the infection. So as this virus is budding off from the cell, the proteolytic activity can cleave that hemagglutinin to make it ready to infect another cell. So it's important to remember this hemagglutination activity. And it gets a little bit confusing from a purely semantic point of view too, because people will confuse HA0, which is the term that's used to describe the uncleaved, the proteolytically uncleaved form of hemagglutinin, into HA1 and HA2, the proteolytically cleaved products. Those get confused frequently with what are known as the serotypes. So I mentioned that there are 16 different types of hemagglutinins that are known. Viruses only have one type of hemagglutinin. Each virus has one type. All 16 types of hemagglutinin will infect birds, but only H1 and H2 and H3 will infect humans. So it gets a little bit confusing when you those terms, but it won't confuse you because now I've just cleared that up. So here's the other thing about why it is that avian flu is, first of all, so severe, and second of all, less likely to spread from humans to humans. So independently, or perhaps in addition to the proteolytic processing that I just described, the typical viruses like H1N1, hemagglutinin 1, neuraminidase 1, that circulate in the human population now, find their receptors, their sialic acid containing receptors, predominantly in the upper respiratory tract. So when you get infected with influenza, typically that infection is limited to the upper respiratory tract. Sadly, for elderly people and small children, they will secondarily get bacterial pneumonia, which is actually lethal. But you and I predominantly survive influenza because it's predominantly an upper respiratory tract. The receptors that allow the bird flu, H5N1, to infect us are not part of the upper respiratory tract. They're down much deeper in the lungs. This is why I think that people need to work with poultry, infected poultry, to get a large infectious dose and to inhale it deep into their lungs in order to cause the infection. So having the infection much deeper in the lungs is much more serious, but also it makes it less likely to spread from person to person because material from deep in the chest cavity is not going to be coughed out as much as it is if it's in the upper respiratory tract, where from the pharynx and the nasal cavities and the throat. So this also probably contributes to the fact that this bird flu is much less infectious from human to human. So in just the last few minutes, I just want to talk a little bit about one of the other really fascinating aspects of influenza. I'm not going to go through this in total, but I mentioned that all 16 of those influenza types infect birds. In fact, birds are the definitive host for influenza A. We're not supposed to be infected by influenza A. Animals, mammals are not. Birds are. So the avian sialic acid receptor has a particular linkage that we don't particularly share. This is also part of the reason why it's difficult for us to be infected by many of these avian influenzas, because avian influenzas, typically those that just infect birds, will recognize sialic acid in an alpha-2,3 linkage to galactose, whereas humans have an alpha-2,6 linkage. So that simple biochemical difference actually protects us to some degree from being infected by these bird viruses. The sad thing is that the poor little pig has got both. Pigs end up being the major mixing bowl. These are the animals that, for the most part, are responsible for the pandemic strains of viruses, because this poor little pig can be infected by a human virus and can be infected by an avian virus, and sadly, it can occur simultaneously. That's the big problem. How much time have I got? I don't have enough time. So now that I've put you on the edge of your seats, when you come back, what day is this? Monday? This is Wednesday. Friday. We'll finish the story on why pigs are bad for influenza.